proud to I'm proud to introduce Dr. Ratna Puri. She has been the past president, and she is a phenomenal figure in the world of genetics. And she's going to take you through the A to Z, actually A, B, and beyond, but A to Z of thalassemia. The whole webinar is structured in a way that we understand the whole concept of thalassemia and the way it affects the fetus. Dr. Aparna Sharma and the whole team of galaxy of expert panelists will take you through practical aspects of thalassemia. Welcome, Dr. Ratnapuri, ma'am. Over to you.
Thank you so much for the kind introduction. And without wasting much time, we will start with the presentation. If you could let me know if you can see the presentation and hear me.
Yes, ma'am.
Yes.
Yeah. So I've been asked to speak on the ABC of and beyond of thalassemia. So I've put together some things that I think which are absolutely important and we should know. And so first is to understand what is this whole thing about thalassemia and hemoglobinopathies. So this is related to any disorder either in the structure of the hemoglobin or in the synthesis of hemoglobin. And it's one of the most common genetic disorders worldwide, especially in South Asia and Southeast Asia. So when we talk of these, there are two main groups that we have to consider, the alpha and the beta. So I should say the beta because that is commoner in India, the beta thalassemias and the alpha thalassemias. And you also have abnormal variant hemoglobins, which are like the sickle cell hemoglobin, the hemoglobin C, the hemoglobin Lepore, hemoglobin D, which includes D Iran and D Punjab, and hemoglobin E. And we'll understand the importance of knowing this in the subsequent slides. I've already mentioned that it's the commonest autosomal single gene disorder globally. It can present either as thalassemia major or thalassemia intermediate in a person who's affected with thalassemia. So this is just a clinical spectrum, and we have specific mutations which can cause these specific phenotypes. So thalassemia major is a more severe form of thalassemia where there is, by the first year of life, there is a drop in the hemoglobin, there is hepatosplenomegaly, the child is weak, not thriving well, and anemic, and then would require 3 weekly, monthly transfusions to maintain the hemoglobin. Intermedia would not present so early. It is a milder phenotype, but really would present at times of stress, if the child is sick or during puberty or during rapid growth and things like that. So they're not watertight compartments. It's a spectrum. And these are the common features that I've already alluded to. So we know that the burden is high. It's estimated that the number of the children and adults with beta thalassemia syndrome is about 100,000. And we have about 10 to 15,000 even today, infants being born with thalassemia major every year. So what do I mean when I say it's an autosomal recessive disorder? I would like you to see this pedigree and hopefully you can see my pointer. This is a family where two individuals are married. In this specific pedigree, they have a common ancestor, and so we say that they are consanguineously married. Both of them are carriers of beta thalassemia. Beta thalassemia, there are two copies of the gene in every individual, and we can see that in carriers, one is the functional gene, the one that's not colored, and the gene that's colored is a non-functional gene. The second thought that always comes up when we talk about a carrier status with anybody is, why is it me? Why am I the one who's a carrier? And putting things in the correct perspective, every human being in this world is a carrier for some gene or the other. And here we know that these two partners are carriers for a mutation in the beta thalassemia gene. They will never be symptomatic because they have a normal gene which takes over the function of this gene that's not working. In every pregnancy, either the functional gene or the one that is not functioning will go to the baby from both partners. And it's like a flip of a coin. We cannot control what is going to go, but yes, we can test to identify if both non-functional genes have gone to the child would be affected. However, even if one functional gene has gone, the child will not be affected, and this includes the carrier state. We normally in pregnancy never talk about the carrier status. We just say that the fetus is affected or the fetus is not affected, because carrier state is not affected. The other important thing that you have to see in this pedigree is that once you identify any carrier, there are other siblings and other individuals within the pedigree who can be carriers or may not be carriers. And therefore, this whole concept of cascade screening becomes very, very important, and at least to provide the information to the family. And we know, because it's an autosomal recessive disorder, males and females are both affected. And for an affected child, the parents would be heterozygous carriers. And I mean, why is it a carrier is not symptomatic? Because 50% of the protein that is formed by the normal copy of the gene is enough to do the function of the gene. And so therefore, when as for any autosomal recessive disorder, if we have carrier parents, in every pregnancy, there is a 1 in 4 chance that the baby would be affected. But this 25% risk of disease versus 75% that the baby is normal is for each and every pregnancy. It doesn't mean that if in pregnancy 1 the fetus is not affected, 2, 3, 4 will be normal. Pregnancy 2 does not know what happened in pregnancy 1. The disease risk is only if both partners are carriers. You can see here that one partner is a carrier, the other is not a carrier. Then there is no risk of disease where we have both red-colored non-functional copies of the gene. The sibling, some offspring can be a carrier, but that is a normal, healthy state. So this is some data from India that our carrier rate varies from 1 to 17%. We say on an average, it's about 3%. There's a lot of work that has been done on thalassemia in India. And we know that the carrier rate is not uniform in some belts. We know that beta thalassemia is common. Tribal population, you have sickle cell that is common. There is a population in the East where hemoglobin E is common, etc. So therefore, it helps to know wherever you are working, what is the common either thalassemia or the hemoglobin variant that is common in that population. The second thing is, why is there so much of thalassemia? Because there have been many studies to show that there is a natural selection. There is a relative resistance of carriers against the Plasmodium falciparum malaria. And therefore, the carriers are selected by nature because Plasmodium malarian parasite is common in India and in Africa. Second is the system of marriages, consanguineous marriages, endogamous marriages, where we all marry within our same community. We have founder effect, as well as a genetic drift of the population. So understanding this concept of globin, why does it happen? I think first, very simply, I'm going to tell you what is the structure of hemoglobin. We have multiple types of hemoglobin in our body, and I'll come to that later. But these are the common adult types: hemoglobin A, hemoglobin A2, and hemoglobin F. So hemoglobin is a structure or a molecule that is made up of iron, which is in the ferrous form, that's the active form, and globin, that's the protein. So you can see that we have 4 units of these proteins. The protein is referred to as the globin, and we have 2 alpha globins and 2 beta globins. So it's like a little butterfly, 2 beta globins, 2 alpha globins, and this is the heme molecule inside. And this is how it functions. So the beta, if there is beta along with the alpha, it is the adult hemoglobin, which is the most prevalent hemoglobin. You can see here, 95% of our hemoglobin is adult. If it is alpha with gamma globin, it is hemoglobin F. If it is the alpha globin along with the delta globin, it is hemoglobin A2. So you have different kinds of hemoglobins within our body, and they have various individual functions. So this is how it really looks like. So this is the heme moiety with the iron inside, And this is the protein that is around this heme moiety, which is holding it all together within our red blood cells. So you can see here that this is the 2 alpha chains, these are the 2 beta chains. Now remember I told you that, there's alpha, beta, gamma. A globin is a protein, and a protein has to be formed from a gene. So we have 2 big clusters within our body. One is on chromosome 11. So this is the beta globin gene. And this beta globin gene goes to form the beta part of the globin, and therefore it is important to form hemoglobin A along with alpha. The alpha gene makes the alpha globin, and this is present on a different chromosome. So essentially, chromosome 11 and 16 are the important. The alpha forms the alpha globin, the beta forms the beta globin. They come together to form hemoglobin A. Next to the beta gene is the delta gene, which forms the delta globin, and this with this alpha forms the hemoglobin A2. And then we have the gamma. There are 2 different gamma genes. They form the gamma globin, and that along with the alpha forms the hemoglobin F. These are other embryonic hemoglobins, and we'll talk to them about here. So you can see that very early in the embryonic period, we have 3 embryonic hemoglobins. They are only present in the embryo before 8 weeks of gestation. It's called the hemoglobin Gower 1, Gower 2, and Portland. And these are they are the primitive. They are very important for primitive erythropoiesis. And of course, they will have different genes. This is zeta epsilon, alpha epsilon. You don't have to remember this, but just to understand that as the baby grows, we have different kinds of hemoglobin. Predominant in the fetus is the fetal hemoglobin, which is alpha 2 and zeta 2. And then this which is alpha 2 and gamma 2, which is the hemoglobin F. And you can see here that the embryonic hemoglobins decline by about 8 weeks. So by the end of the first trimester, there is no embryonic hemoglobin. And as the embryonic hemoglobin comes down, the hemoglobin F increases because alpha is being produced by the alpha gene. And when alpha and gamma come together, they form the fetal hemoglobin. This should be gamma. I'm sorry for this error. And then after birth, the hemoglobin F comes down, the fetal hemoglobin starts coming down because the baby is no longer a fetus, and the adult hemoglobin comes up, and therefore the synthesis of the beta globin moiety increases. So giving us then predominantly hemoglobin A and some amount of hemoglobin A2 as the delta gene also kicks off. Now, this is what the structure, I thought I would want to introduce everybody to what a gene looks like and what does it do. And this is the simplest gene we have in our body. This is the HBB gene. Okay, this is the beta globin gene, the commonest gene that is sequenced in beta thalassemia. So it has 3 parts, which are the coding parts. They are called the 3 exons. There are some control regions. Okay, it's suffice to say that these are all control regions ahead of the gene and after the gene. Most of the mutations lie here, not in the introns, but in exon 1, 2, and 3. There are some mutations which can lie even in the control regions. So therefore, we know what the mutation profile is, and so we also need to sequence the control regions along with the exon 1, 2, and 3. So we said that thalassemia syndromes is inheritance of an abnormal beta thalassemia gene from both the parents, or one abnormal beta thalassemia gene along with a variant hemoglobin from the other parent. Both these can give a phenotype of thalassemia. Why does it happen? So what if the globin chain is not being formed? It's important to remember that in the body, the alpha globin and the beta globin are in a 1:1 ratio. Any point in time where this ratio gets disturbed, supposing the beta gene is not working, the beta globin chain is going to be low. So there's going to be more alpha, less beta, and it's going to precipitate. and it precipitates in the RBC precursors. And because of that, it injures the cells, there's hemolysis, the hemoglobin falls, then the bone marrow becomes hyperactive in an effort to maintain the hemoglobin. And then we get the entire phenotype of beta thalassemia because of extramedullary hematopoiesis. So you can see, I've already talked to you about this. So we can have beta thalassemia, major, intermediate, or carrier. Carriers or minors are not symptomatic. Sickle cell disease. We can also have alpha thalassemia, which will occur when the alpha gene, remember on chromosome 16, has a mutation and does not form the alpha globin moiety. And if the alpha globin is not formed, alpha decreases, beta globin increases. Again, that will precipitate and cause disease. And I'll come to alpha thalassemia later. And I've already alluded to this. It's important to know you can have just both mutations in the beta globin gene. You can have symptomatic alpha thalassemia, and you can have sickle cell disease where either both mutations are causing sickle cell disease, or there is one sickle along with beta thalassemia, or hemoglobin D or hemoglobin C. These are the various kinds of variant hemoglobins. And always to remember that a carrier is not symptomatic, and therefore you have to proactively do the test to identify a carrier status. So, absence of the beta globin gene synthesis, there are two types. If there is the beta globin gene and that gene has a very severe mutation, it will not produce any beta globin chain, and that is the beta 0 type of thalassemia. If it is a milder mutation, it will produce some amount of beta globin, but at a reduced rate, and that is a beta plus type. Both are carrier states, remember. And it is dependent upon the kind of mutation and the place where the mutation is in that beta globin gene. So if you have two beta zeros together, there is absolutely no beta globin chain synthesis, and you get a severe phenotype. But if your mutation is of the beta plus type, there would be some production and you would get a milder phenotype. And how do we know whether it's beta 0 or beta plus? Because I told you a lot of work has been done on thalassemia in India. So based on the nature of the mutation, you are able to predict whether it is beta 0 or beta plus. How do you identify a carrier state of beta thalassemia? We do the HPLC hemoglobin A2, and I think it's absolutely important that along with just the A2 level, you can see that this is normal. This is the predominant hemoglobin A. This is a normal A2. This is something that comes up in this window. So this is abnormal. Okay, so this is, I think, hemoglobin S. Yes, this is hemoglobin S. Then you can have a very, very high hemoglobin A2. And we know that in this window, along with A2, there is hemoglobin E and hemoglobin D Iran that elutes. And this is again what we refer to as a high delta beta thalassemia carrier. So these are the interpretations of the HPLC. And along with this, it's important to note that we must have the hemoglobin, RBC count, MCV, and MCH. Please do not interpret this individually. And I'll give you a case examples to see why it is important. Because if you look here everywhere, they talk about, is the MCV, MCH normal? Is the MCV, MCH reduced? So these parameters become very important. So we've already talked about the beta 0, beta 0. So if both mutations are beta 0, beta 0, or beta 0 and a severe beta plus, it's going to be a very severe thalassemia major. This column, the third and the fourth column, would be milder disease because the mutation type is milder. And then you could even have much, much more milder. So it's over a spectrum. And this genotype-phenotype correlation becomes very normal. Because sometimes if the family plans that they want to continue, you would want to guide them as to what is the nature of the mutation and what would be the disease phenotype that the child could have after delivery. And we know that this is the carrier status and there would be no disease phenotype. Now, what are the common genotypes that are associated with a clinically significant disease? So every combination is not associated with a disease phenotype. And you would want to do prenatal diagnosis only in these situations that you can see up here, because these are associated with the disease phenotype, except for number 4 here, which is a combination. So this is beta thal carriers, both of them, beta 0 or beta plus. Now this should make sense. There is another hemoglobin variant, which is called hemoglobin lepore, along with the beta thal carrier, can cause severe disease. Delta beta thalassemia is a kind of deletion thalassemia, and that with beta thalassemia trait will also cause disease. But where you have hereditary persistence of fetal hemoglobin, your hemoglobin F will be very high, more than 50%. There will not be any disease phenotype, so you will not do a prenatal. And whereas when you have E beta thal, again, you need to do a prenatal. And the same is here for sickle cell, sickle with thalassemia, sickle with hemoglobin D, sickle with hemoglobin C. All these situations require prenatal testing. These are the conditions, and I think it's very important to know this, where they are not affected with a major disease. These are asymptomatic states. Homozygous, the common ones, I'm just going to say it, homozygous E. If you have hereditary persistence of fetal hemoglobin, heterozygous or homozygous. If you have hemoglobin D with beta thalassemia, any form of hemoglobin D, D Punjab, D Iran, you don't need to do a prenatal diagnosis, whether even if it's a hemoglobin D with hemoglobin E. But when you have a D with S, you will need to do a prenatal diagnosis. So it's important to know these conditions. We know a lot about the mutation spectrum in our country. We also know that in different parts of the country, we have different mutations. Worldwide, there are more than about 300 mutations that are known, but we have common 5 and common 10 mutations in India. And suffice to say that most of the times we look at this, and if these mutations are not there, only then you can go ahead for the full gene sequencing. And these are the common mutations that are present in the country, also showing you which part of the country, what mutation is commoner. But you can see that overall, this IVS15G2C is the common mutation all over India. Coming to alpha thalassemia, in alpha thalassemia, we would now know that there is a deficiency of the alpha globin gene because of the alpha gene that has mutation. Alpha genetics, alpha thal genetics is a little complex because we have 4 genes, not 2 genes for producing the alpha globin. So we have again a severe form of alpha thalassemia, which you can see here. The most severe form is this, where it is hemoglobin H disease, where none of the genes are working. This usually presents with hydrops fetalis, whereas the less severe form is hemoglobin H disease, which presents postnatally after birth or the first, second months of life, where 3 of the 4 genes are not functional. Whereas the other situations where 2 genes are functional or 3 genes are functional, that is not a disease state. So before I conclude, there are just a few things that I thought I would like to reiterate. Who to test? So we know that every pregnant lady, whether she in her first visit, not because her hemoglobin is fine or her hemoglobin is low, you decide first you'll give iron. First visit, every pregnant lady must have a CBC and a HPLC-A2 or in the preconception. It's important that we take a family history so that if there is a family history of thalassemia, we are already alerted to it, but we need to test it even in the absence of a family history and also for gamete donors. And if you have if you've done an HPLC in the first pregnancy, please don't repeat it in the second pregnancy because that will not change. So we know we have to test it as soon as possible. What are the red cell indices to alert us to a carrier state? A mean corpuscular volume, MCV of less than 80 femtoliters, and MCH of less than 25 micrograms. If you have an RBC count which is relatively high for the hemoglobin, if the hemoglobin is 9 and your RBC count is 5, it should alert. It should alert you. There is a little, a small percentage of what we call silent carriers. That means they would have normal MCV, normal MCH, a normal RBC count, and yet they may or may not have a normal hemoglobin A2. Yet they are carriers when we do the mutation. So there is data, and a lot of data has come from the National Institute of Immunohematology and the group which was led by Dr. Roshan Kola. Now there are other people there who have published and told us that the silent carrier state is prevalent in India. And if one partner is a carrier and the mutation identified, and the other partner has a normal indices and a normal hemoglobin A2, it may be beneficial to do, in fact, they say you should do it. You should do the mutation profile of the partner so that we don't miss a silent carrier state. What is the HPLC that suggests a carrier status? Normally, the normal hemoglobin A2 varies in different labs. 3.9 was the cutoff that is traditionally used. Anything more than 3.9% is a carrier. However, the silent carrier varies between 3 to 4%. So that is your silent carrier zone where there may be carriers. What are the pitfalls in carrier testing? Normal RBC indices will be present in carriers. So there are situations where you can be a carrier, but why don't the indices show that you are a carrier? Remember, everything is because of an alpha globin gene mismatch. Now, if my beta globin gene is low, but supposing my alpha gene is deleted, alpha is also low, beta is also low. So that will give us a normal indices, which will be a false negative. Okay, so that's why co-inheritance with any alpha gene mutation. So an alpha thalassemia carrier are also microcytic hypochromic, but they will have a normal hemoglobin A2. That is when we suspect an alpha thal carrier. Silent beta carrier, I've already talked about. They have normal indices, may or may not have a normal A2. They can be within the borderline zone. And then these are other slightly complex situation where you have microcytic hypochromic anemia, but we have a normal hemoglobin A2. So first thing about iron deficiency anemia, common in India and can make the hemoglobin A2 normal, but it really has to be very, very severe for it to alter the hemoglobin A2. Co-inheritance with alpha thalassemia, I've already talked about, as well as other types of thalassemia like delta beta hemoglobin lepor can alter this status. And I'm going to conclude my talk now with 3 cases that are very important. This one is to talk about the importance of cascade screening. This couple is pregnant, knowing that there is so much of family history, a sibling here and a cousin here. But nobody talked about the 50% chance that each one of them could be a carrier. So when you identify one carrier couple, it is our responsibility to educate the couple about testing the carrier status in their at-risk family members. Okay. Then I come to this. When you have a situation where one person is a beta thalassemia carrier, whereas the other individual is a hemoglobin D carrier. I'm sorry that this has gone off in the color, but what I wanted to show you here was the CBC, MCV, MCH, RBC count is normal. In this situation, we would think that this is hemoglobin D. You confirm it by the molecular method. There is no beta globin gene mutation, which you would not expect in a routine because the CBC is normal. There is no indication of offering prenatal diagnosis in this pregnancy because even if these two come together, they do not cause disease. What are the prerequisites for prenatal diagnosis? The partners should be carriers, mutations must be identified, the couple counseling should be done, and when you do the mutation, you just check the targeted mutation in the fetus. The mutation of the mother, the mutation of the father is what is tested, and the rest, of course, there is pre-test and post-test counseling. Now, this is an interesting case where the wife was a carrier and the husband's A2 was reported to be 3.7, and this is how the report came. They came to Gangaram for a prenatal testing. So when we started looking for the mutation, we identified the common mutation in the lady, but in the husband, we could not. Okay, we sequenced the whole gene, but we could not. And so we repeated the CBC of we didn't repeat, it was not done. The CBC parameters were normal. So we wondered whether the husband was a silent carrier. Anyway, he was to come to India. We repeated everything in our hospital. The CBC was normal, the hemoglobin A2 was normal, and then he was not a carrier at all. So therefore, before you put in the needle and you do take out a CVS sample, make sure that all the mutation profile is done. And you also look at the CBC and you look at the hemoglobin A2 levels to ascertain that, your partners are carriers. So you have to look at the entire picture and not just go for prenatal testing of the fetus. And this is the last case and very important because this is a learning case. So this was a couple who were pregnant. The wife had a high hemoglobin A2. You can see here 4.7. Her MCV was low. Her MCH was low. Her RBC count was high for a 9.5 hemoglobin. So she was a sure shot beta thal carrier. The husband was reported to have an unknown peak in hemoglobin Q. This is a variant in the alpha globin gene. Now, if he's a hemoglobin Q India and the wife is a beta thal carrier, there is no indication for prenatal diagnosis. But what do you have to see here? You have to see that there is microcytosis, there is hypochromia, and look at this RBC count of 7 for a hemoglobin of 13.5. Even though the hemoglobin A2 was reported as normal by the lab, this should alert you. Either the patient has iron deficiency anemia, it's unlikely because the RBC count is very high. This patient was actually had hemoglobin Q India and was also a beta thalassemia carrier. And this is, remember I told you, these are the borderline hemoglobin A2s. So if you miss this, you miss a very, very major thing that both partners are carriers and the fetus would require prenatal testing. So to conclude, I think prevention is the key to decrease the burden. You have to screen every woman and couple, and I think we have to spread the information. Whilst we've been talking about this for many, many years, we still haven't achieved it. Please do a complete blood count, which includes an MCB, MCH, RBC count, and hemoglobin, along with an HPLC, hemoglobin A2. The family history is important. Ascertaining the risk in the cascade screening is also relevant. Prenatal diagnosis to prevent burden of disease and prevent the birth of affected children, and must identify the mutation before putting in a needle to take out a fetal sample. And the 25% risk is in each pregnancy and not for 4 consecutive pregnancies. Thank you very much.
Thank you so much, ma'am. I don't think I've ever heard such a comprehensible, sorted, easy to understand, lecture on thalassemia ever.
Thank you.
Wonderfully given. Thank you so much. And ma'am, I would also like to tell you that on behalf of Society of Fetal Medicine, we have done all the chapters. I've in fact done some outreach information, in different way to reach out to the public to create the awareness of thalassemia on this day. So some have given it on TV, some have given it in as local ad in newspapers, and probably this is a humongous effort which I applaud all the chapters for carrying out on today. So with this, I let's have the trade videos before we move on to the panel discussion. Thank you so much again, ma'am. Vishal, can we have the trade videos?
Yes, sir. Atul, play the SFM video, please.
Give me a second.
Now I invite upon Dr. K. Aparna Sharma. She's associated with AIMS, the fetal medicine unit of AIMS. She is absolutely clear in her thoughts and concepts, and I request her to take us through her panelists and the panel from thalassemia from policies to practice. Over to you, Dr. Aparna.
Thank you, Dr. Mohit. And thank you, SFM, for observing the World Thalassemia Day today. I think this is a very, very important occasion, a momentous occasion that and the activities that are being held under the banner of SFM, the messages that we are trying to deliver today. And I'm extremely honored to have the eminent panelists here on the panel today, leading with none other than Professor Ashok Khurana, sir. We have Dr. Sumita Ghosh, ma'am, Dr. Madhulika Kabra, Dr. A.G. Radhika, Dr. Suman Mendirata, Dr. Hemchandra Pandey, and Dr. Mukul Agarwal. We all know Dr. Ashok Khurana, sir. He is the mentor emeritus of Society of Fetal Medicine. We don't need to introduce him, but I would like to say that the panel today is his brainchild. He said that we need it to be in a way that is not just academic, but we also need it to be practical from actually the practice to the policies on what is actually happening on the ground. So that's why we have put together a panel, which is, I would say, who are the movers and the shakers who are actually working in this field, who are dealing with these patients and who are trying to bring about a change. We have, of course, with us Dr. Sumita Ghosh, who's currently the officer on special duty to the Niti Aayog, who was the ex-additional commissioner in charge for child health, adolescent health, and safe abortion services. And for us as a community, she is most well known for bringing about the much needed change in the MTP Act. So welcome, ma'am, for being here today. We, of course, know Dr. Madhulika Kabra, ma'am, who is the professor and head Department of Pediatrics and in charge of the Division of Genetics at AIMS, New Delhi. And ma'am has been leading the prenatal diagnosis for in this field for a long time now. And welcome, ma'am, to this panel. And we really are looking forward to your inputs on this topic. We also have with us today Dr. A.G. Radhika. Madam is Senior Consultant OBGYN at the UCMS and GTB Hospital. Apart from being a great academician, she's also worked a lot in the field of thalassemia right from the very beginning in the early 2000s. So she has had a lot of experience in this field, and she was going to share that experience with us. We also have with us Dr. Hemchandra Pandey, who is the additional professor at Department of Transfusion Medicine at AIMS, and who routinely handles the patients of thalassemias requiring regular blood transfusion. And Dr. Mukul Agarwal, Associate Professor of Clinical Hematology at AIMS, who manages thalassemics on a routine basis. We also have with us Dr. Suman Mendirata, who's the Senior Gynecologist and Head of the Department of Hindu Rao Hospital. Hindu Rao Hospital has been the center for managing screening thalassemia in Delhi for the last almost 2 decades now. And she's going to share her experience for screening thalassemia in the hospital and in Delhi. And I would also like to acknowledge Dr. Jyoti Sajdev, ma'am, who's State Program Officer for Maternal Health, who has really given the inputs for how the program has been running in Delhi. So with such eminent panelists, I would like to take this panel forward with some of the basic questions first, which I would like to begin with Dr. Khurana, sir. So in terms of the burden of thalassemia, sir, it's very easy to say the numbers that so many thalassemic children are born every year, but how heavy is the burden of thalassemia in India, and why should we be worried as obstetricians, as radiologists, or just healthcare providers at the forefront? Why should we be worried about thalassemia?
Thank you. Thank you, Aparna. It's a privilege to be here this evening. And it's been a year since we celebrated World Thalassemia Day last year. And today we're using Thalassemia Day to also look at the rest of the hemoglobinopathies. But first things first, we keep saying how heavy is the burden of thalassemia. And we need to change our approach to this whole question. It is not the burden of thalassemia, but the responsibility of thalassemia that you and me need to think of. It's not a burden. It's something that we have allowed in society. It's something that we should not allow in society. And therefore, it becomes definitely the responsibility of a disease that can be prevented completely, because we have those examples in history. In India, if you just take the beta thalassemia, we heard from Dr. Ratnapuri that there's 3 to 4%, that it's much more in certain communities. We have the Punjabis, the Sindhis, the Gujaratis, Bengalis, Mahars, Kolis, the standard group that we always give, the Lohanas, the Gaur Brahmins. All of these are all included in this, and the Saraswats. And then if we look at just hemoglobin S, we know that the tribals from southern India, western India, central India have an incidence of as high as 48%. And this is all part of the survey that was published in 2016, and it's probably more now. We've had hemoglobin E reported from the Northeast states as high as 50% carrier state. And of course, hemoglobin D in Punjab is 1 in every 50 Punjabis have hemoglobin D. So we really have this huge incidence, which is why we have 10,000 to 15,000 babies born every year with Thal major. Of these, of course, half will not survive beyond the second decade. But essentially, we have about 1 lakh patients there. Now, let's see what happens with them. The answer, of course, is a bone marrow transplant, or you can have fancy names for it. But since that is not available everywhere and it's expensive, we're stuck with transfusions and with chelation. So when we come to chelation, let's look at what Israel calculated just a few years ago. They said, look, there's $2 million for average life expectancy, and there the thalassemics seem to survive till about 50 years of age. And so they said that for 1 year, it's like $400,000. Now, I'm not sure how they calculated that, but closer home, if we realize that you take a 30-kilo average baby, 1 year, it's about 2 lakhs for the chelation and the transfusions. That's a huge amount of money for the family or for the state to spend. And if we add that up to our 10,000 kids, we're spending 10,000 crores per year to look after these children in India. Now let's compare that with trying to institute a program where we identify these people, where we give them the parents on whether they'd like to have a baby with all of this and with the possibility of a bone marrow transplant, or would they like to have information and make appropriate reproductive choices. And we worked this out in Uttarakhand a while ago, and we realized that if we take just the cost of testing, HPLC plus the positive cases, and add the mutations testing to it, about 1,500 would be spent on an average. And this means that we actually weren't spending very much. It's not even equal to the cost of that program for even 3 months. So that means that we really must intelligently spend our money. And it's been proven again and again. In Cyprus, we know, and we hear this from Kypros Nikolaidis every year, what they did was if they didn't have their program in place where they look at school children and young adults and screen them, we'll actually have a 600% increase in blood transfusions within 10 years. Nobody can afford that. And we've had good examples from Montreal, we've had good examples from Latium in Italy and so on. And therefore, you and me are left very simply with the need to definitely screen. Yes, we did attempt to definitely screen a lot of school children and young adults in the early 90s. And these girls got labeled and they never got married. And I know that from the Punjabi community that we had. That was what was called Jamna Par in those days and is East Delhi now. So there is that aspect of getting labeled with the disease. But does that mean that we'll allow people to suffer? So this is what I hope we will be able to sort out tonight and realize that our policy needs to be accelerated because there's a lot of money here that is going waste. We have to use that intelligently.
Right, sir. So I think that's very well put, and I think the stigma around the screening has to be something that we need to talk about. That this is not something that is that's going to be there, and it is preventable, and we just need to not propagate it further. So I think that with that we would like to now ask Dr. Radhika that what is her experience, that can we actually eradicate thalassemia? What has been the worldwide experience, and what has been her experience? Because working in GTB for so long with the thalassemia screening programs, what is the experience that you would like to share with us?
So, good evening, Aparna and everybody. Thanks for having me here, SFM team, SFM and Aparna. I am kind of intrigued by this question. Can we eradicate thalassemia? Of course, yes. So the point is, we need to start with a positive frame of mind, and of course, we need to get together to do this. I think people all over the world, I think we all know about Cyprus and how they have done it. We've been watching them, and of course, you've given all these statistics here. But I think in India, we still need to go a long way. There is a huge diversity of practice. If I just consider Delhi, so the variation is too much, and so it is for the rest of country. So now we have come forward, we have moved away from mortality statistics to morbidity. That is a big change that has taken place in terms of improvement of the healthcare in India. So all this while we were trying to bring down the mortality, the maternal mortality, the neonatal mortality, and so on and so forth. Now we have been to a large extent contain, or at least bring down the figures. So today we are in a position to talk about other aspects, and that is something very important, the hemoglobinopathies. The focus earlier was the school children. A lot of studies had been done today. In the past about 20-25 years, Delhi government, I think, was the first one to initiate the thalassemia antenatal screening program. And it in Delhi started in 3 hospitals, and that was the GTB hospital, Maulana Azad, and the DDU. These were 3 hospitals.
Right.
So at our hospital, that is GTB, it started in 1999. So, of course, there are the teething problems. You start with having the people, the right kind of, they had allocated a manpower, medical officer, lab attendant, and a lab technician. So, we all know how it is to get the right kind of people. But we started off very well. So, the mandate was to screen women, the primaries, less than 16 weeks of pregnancy, equal to or less than 16 weeks. And after counseling, initially it was a Nestrov test. So screening had to be mandatorily done, followed by HPLC, because in those days we had only one machine. So we had to contain the numbers. So it went on very well for at least about 20 years. And we kept on projecting the figures, and our work was well appreciated at all levels. But then there was this thought that since it is running so well, why don't we make the staff permanent? Okay, so the file started moving in the year 2019. So immediately that was just before COVID, then the COVID stuck. So there was a halt in that movement of file, and then everything just kind of collapsed. People in the planning did not understand what was wanted. So now, if you ask me today, it's just come to a standstill. We till 2023, the entire staff was got removed. So somehow we cajoled the hospital administration. We have a medical officer now posted, and we restarted in 2023. So this is the overview of our struggles and our processes to keep the program running. And because innumerable number of court cases, because of the people who got terminated, they were so disgruntled, they could not find alternative engagements because they had crossed the limit of age to apply for a job. So that is the level of, the problem that it can happen. But in terms of detection, if you really ask me, so in so many years, how many, the total numbers, I would say more than about 80,000 couples have been tested. The prevalence of thalassemia trait is to the tune of 1 to 2%, which is, in terms of percentage may not be high, but in terms of number, we know it is very high. And the couple was about 0.2 to 0.8%. So what we have been doing is after, till that stage, because we didn't have a fetal therapy unit, we didn't have a fetal medicine unit at our place. So initial referrals were getting sent to AIMS, and then in the recent past, it's going to Maulana Azhar. So we used to have a good feedback mechanism between to and fro between AIMS and us, and also the couples used to report back to us. So till such time, we had a sort of feedback to know the number of pregnancies, the number of positive fetuses that we could, successfully avoid the deliveries of, and that was about 30 fetuses which we could avoid their deliveries. In the recent past, that is since 2023, not only we have been testing for thalassemia, we have done away with Nestor because there is no lab technician now. Then there is a limitation to the number of samples that can be taken per day, that is 10. However, despite all that, we have also been testing for hemoglobin D, E, S, and Q. So the prevalence for HBD is about 1.4%, which is more than thalassemia, beta thalassemia. E is 0.5, S is 0.3, and Q is 0.15. So this again was an eye opener. So how we got this moving was now the people have started doing thesis. So this has ensured that there is a resurgence, there is a life which is breathed into the program. So because of that reason, we have also been able to identify these other variants that are there. So, however, if you ask me in the last 1.5 years, how many fetuses were positive, I'm not able to share that information at the present point of time because the medical officer was posted has now been shifted out, and we presently do not have a good feedback mechanism. So, that's how the nutshell of the entire program at our place.
Right. So thank you, Dr. Radhika. I think first of all, first thing is that you've screened 80,000 women, and I think that really tells us the numbers and the effort that is going in. And the screen
80,000 primaries,
yes.
less than 16 weeks.
Yes, so primary is like without affection, and I think that is talking about the kind of work that is being done. So I think that's extremely important, and also you bring about the challenges. It's important to understand that it's easy to say that we are going to do it, but at the end of the day, it really needs a huge amount of effort to sustain it. So thank you, Dr. Radhika, for bringing that point out. And I would also like to point out that the Indian government actually has the existing screening program for children, the Rashtriya Bal Swasthya Karyakram. But if you look at this small point here, the point number 30, which actually points out that you have 30 conditions which needs to be screened, where beta thalassemia is optional. So these are some of the things that should be raised, that why we have to keep it optional when so many other things are mandatory. So I think these are some of the questions that really need to be raised. Yes, Dr. Radhika.
So, Aparna, there is now the special, the Ministry of Health. So they have started under the NCD programs. There is something on the hemoglobinopathies. So I think people are slowly waking up, and this is an answer to your question whether we can eradicate it. So
Right.
I have a very positive outlook in that direction.
Right. So you wanted to say something about this?
Yes, so I totally agree with you. I don't like thalassemia being placed as a footnote sort of a thing, and that
Yeah.
too optional. Just not that's not the way it should be.
Yes.
And but the heartening fact is that the ministry is now also
Yes.
gearing up.
So I think this is something that should be mandatory, and there's no reason for it to be really optional in this sense.
Absolutely.
Yes. So Dr. Khurana, just coming back to it, like you already have touched this point a bit in saying that, when the school program was voluntary, when the school program was initiated, a lot of girls were stigmatized. So I think the screening program or anything has to be seen in the totality, the morality of screening versus and the technicality of screening. So the questions do come across that when we screen, are we infringing on the personal rights? So what exactly is ethical screening, and how do we actually ensure that it's a voluntary genetic testing? So this is not to say that, we should not be, of course, we have to be very, very proactive on doing it. But of course, there is this part of the society which always we are living in a society that we are living in. So we have to be very mindful of it. So how do we go about it? Just a few words about this thing.
So to put it very bluntly, when we did this program in the early 90s, society was different. It's very easy to stigmatize somebody, and not everybody was marrying everybody else. And therefore, the choices for marrying somebody were very limited. Today, luckily, at least in the urban background, we have a lot of choices on whom to choose as a partner. And therefore, I don't think that the experience will be so bitter as the one we went through 30 years ago. And so, because the status of women has changed so much, particularly in the last few years, we understand that it may not be so severe. And therefore, I think that this voluntary genetic testing is something we will need to opt for. And parents would actually opt for this because they would realize that it's better to have a woman not get into a marriage where she will run into trouble rather than a girl coming back because something went wrong with that marriage for this reason. So they'd rather be forewarned, and most girls would then make a career for themselves and then get into a marriage where they realize there will not be problems or there will be options. So it's now really a outmoded belief that it cannot be done. It can be done.
Right, right. So I think this is extremely important. Like we are moving in a society which is forward looking, and people do realize their rights. And of course, we need to offer it, but at the same time, it should be universally available. But of course, it should be a voluntary testing. Dr. Radhika, you have a point.
Yeah, so we've been having these presentations, many times, especially 8th May has been the Thalassemia Day, so many years. So at the end of presentations, I generally put on that slide which says match the Janampatri, of course, yes, but also match the Thalassemia status. So
Yes.
that was how Cyprus could do that, because they also engaged the religious leaders,
Yeah.
the cultural, they got it. They went deep into the society. So
Right.
so it should be with us, and I think that's a very. I thought that was on a lighter note, but also important.
Right. Thank you. Thank you for bringing that out. So now we move on to the next part. We just want to know from Dr. Suman Mendiratha, and I'm extremely delighted that she has a publication in our very own journals, the Journal of Fetal Medicine. So I would now invite Dr. Suman Mendiratha to talk about her experience of screening for thalassemia. And over the years, she's like, they have looked into a number of patients. They have some data on awareness as well as their screening percentages. So Dr. Suman, welcome to the panel, and please do share your experience.
Very much. I thank Fetal Medicine Society for having me here, and Dr. Ashok Khurana and Dr. Aparna. So I've been working on this subject for almost 2 decades. So first of all, I just want to share about the level of awareness. The level of awareness, this I have done a study in 2016, and the level of awareness about thalassemia major was just 6%. It was in my OPD. uh, I did this study, OPD area, and for thalassemia state, it was only 1%. In thalassemia major, most of the patients, when there was a thalassemic birth, then only they came to know about this. And in that source of knowledge, also I have seen the source of knowledge was only family history in 15 patients, that is 3%, and through media and publicity in 2% only. And about thalassemia trait, the awareness is still very poor. Now, my article has been published, and it was thesis done on DNB thesis done on this subject, screening of women in antenatal period for thalassemia carrier status. So in this, I've seen, like we have been talking how to screen. So it is very important to screen whatever way you can. Every hospital is not having HPLC or CBC. In periphery, in MCD, I started this screening program, and it was started in 2008. So we started screening school children, and in maternity home antenatal patients, we started screening, and we started with Nestroff only because there were no this thing CBC facility available. So we have done with Nestroff. And then in this study, we have compared sensitivity, specificity, positive predictive value, and negative predictive value of Nestroff, RBC, MCV, MCH, and RBC. And here we can see the sensitivity is 78, 73, and 73 for MCV, 60 for MCH, and RBC count, it was 32.91. And the specificity was 94.14, 70.25, 85, 0.88, 74.16, 93.181. Then positive predictive value of Nestor was 53.45%, RBC count it was 17.47, MCV it was 30.8. So positive predictive value of Nestor was quite good. And then negative predictive value of Nestor test was 98.08. And for RBC count, it was 96.86, 97.41, and 95.66 with MCH, and RBC count, it was 9.422. So here I just want to emphasize that screening can be done even with Nestor test, because blood cell counters, they are not available everywhere. So now I've heard that they are like the program has been started and they're saying it should be done CBC, but blood cell counters are not available everywhere. So we can do this screening with Nestrop, and I have detected quite number of patients. And I will tell you, once I used to visit schools also when we started this program in schools. There I have families, like four members in a family, both parents and both children, they were trained. So this screening was done only with Nestrop test in the schools. And somehow we could not continue it. Our school health schemes, school centers, they were merged with MNCW only. So now it is not possible, since they were not having any lab technicians there. I used to send lab technician from hospital sometime for screening camps and all. And now this program is not running in schools. Now we are doing only in maternity centers. There were 33 maternity centers earlier. Now our maternity centers, also due to the shortage of staff and non-availability of resources, now centers, we have only now 11 maternity centers. And we have 4 major hospital, 11 maternity centers, and I am getting referral from all these hospitals. So this one of my Aparna, can you show me the previous slide? No, before this. Yeah, this article was published in International Journal of Reproductive Contraception, Obs and Gynae, and in this I have shared my 5 years experience. And in this, we have screened around 93,871 patients were screened. And out of these patients, the screen positive patients were 10,983. And hemoglobinopathies, various hemoglobinopathies were detected in 777 patients. So out of these 777 patients, the various hemoglobinopathies were beta thalassemia trait in 82.4%, and prevalence was 5.84%, HBE in 79%, HBD in 5.4%, and HBS in 2.05%, and 1%. And now in antenatal patients, there was 7,737 antenatal patients, and total couples who were positive was 42. Only 16 opted for genetic testing. Rest of them refused for some reason or the other. And out of these 16 patients, CVS was done in 8. And in 3 patients, it could not be done because of late pregnancy. And in the 5 patients, it was not required, they were told. The 3 major thalassemia major fetus were terminated by this screening program over the period of 5 years. Next slide, please.
Right, ma'am. So, yeah, so can we just skip this part, because I think this will come in the PND also. So
Okay.
can you tell about...
Can you just show me this slide, this pie diagram? I just want to emphasize that I have calculated in our setups MCD, only 18% patients we are seeing in major hospitals, and rest of the patients, 52% in maternity home, 23% in school, and 7% in colony hospitals. So if we are just screening in hospitals only. If we really want to prevent or eradicate thalassemia, then we have to start screening in peripheral institute. If we just keep on doing in hospital, it is not possible, because we are covering 18 to 20 patients.
Right. So I think these are some of the IEC activities that you...
These are, I just this I have prepared IEC material for public awareness. These are some posters. These are pamphlets we are distributing. These are some posters we have made. And participation of community is very important, because in antenatal clinic, when I see some lady identify some active people, and I train them, and then they are training their like they are propagating the knowledge in their nearby areas. And in sometimes she comes to hospital also, and she tells other patient about thalassemia prevention. And pre-marriage screening is very, very important. And these today, Thalassemia Day was every year I'm doing these activities. These posters were made by my DNB students. Today itself, we had program in our hospital, and then we had awareness program for students, medical students, general public, and other staff members. So I think all should do this activity. This is how we can generate awareness and can prevent thalassemia. Thank you.
So thank you very much, ma'am. I think this really shows that, the amount of effort, but the important thing also is that, you've said that only 18% of the women are actually coming to the hospital. So it is a general idea that we are already knowing, it is kind of
No.
fact.
We are covering only 18% the
Yes.
patients who are coming to hospital.
Yes.
Rest 72%, they are being seen in peripheral institutions.
Yes,
So if
definitely.
we want to prevent thalassemia, then we have to start screening in those in
Okay.
small centers also.
Right. So I think
we are doing screening, like I am getting referral. We have HPLC machine in Hindurao Hospital.
right.
It is under gynae department only. The one thing I have kept this machine under gynae department
So
only.
I think this
Is
is
a
great.
there is a pathologist attached to this thing thalassemia program, and we are doing antenatal screening in hospital itself, and we are getting referral from other hospitals, Kasturba, GLM, and then other maternity centers, maternity homes. They are doing screening. Some of them are doing screening by Nestroff method, and they are referring positive patients to us.
Right, right. So I think a great sharing of experience, ma'am, and I think it really shows that being passionate and you can really make this happen even in the government setups. So we just have to kind of push it, and like Dr. Radhika also said that you can make it happen. There are challenges, but you really have to push it through. So now we move.
The problem is staff. We
Yes,
are not getting technical staff.
Problem, but yeah, we do.
As I told you, we started in schools also. The problem was they are not having regular lab technicians.
Definitely.
So it is not possible.
And
we can go once a month or twice a month. Otherwise, it is not.
Dr. Sumita Ghosh who's listening. So I think it is going to the right ears that the people are there. They are trying their best, but we need help. So I think that's what the message that we want to pass on, that it is important to do. We are trying, but the things need to be done. So moving on to the next topic, that is prenatal diagnosis. It is important to screen, and how can we prevent? So a lot has been covered regarding the genetics. And for this, I would like to invite Dr. Madhulika ma'am. But before that, I would just like to share one small experience for 2 minutes. What really happened is 2 days back, I was doing a CVS for a patient, and I just got talking to her. She has an 18-year-old child who is thalassemic. So I just got talking to her and just learned how she has been through these 18 years of her life has revolved around her child. And the questions that I asked and the answers that I got really got me thinking on why we are not doing more. So just to share this video for half a minute with her consent, I have taken this, and I think this is something that we need to do.
तो अब आपको इधर उधर आप कहीं मत जाइए, सीधा जीटीबी हॉस्पिटल जाइए, वहीं पे इसका सब ट्रीटमेंट होगा। और फिर हम इमीडिएटली वहीं लेके गए, और फिर वहाँ उसको एवरी मंथ बोला कि ट्रांसफ्यूजन करना ही पड़ेगा। और कोई 1 साल के बच्चे को अपने सामने ये सब होते हुए देखना, आप सोच ही सकते हो एज अ मदर। बाकी आप बोल रहे थे कि कोविड के टाइम पे वो तो अभी 2-3 साल पहले की बात है। बाकी 18 ईयर तो हो ही गए ये सब देखते हुए। तो इनफैक्ट मैं इस प्रॉब्लम को लेकर के डिप्रेशन में भी चली गई थी कि हमारे साथ ही क्यों। अदरवाइज ऐसा बोलते हैं कि जैसे थैलेसीमिया सेम फैमिली में या कहीं सर्कल में कुछ होता है तो चांसेस ज्यादा रहते हैं। मेरे हस्बैंड आसाम के हैं, मैं पंजाब की फैमिली से हूँ, तो भी हम लोगों का हो गया कि हम लोगों को अवेयरनेस नहीं थी इसके बारे में उस टाइम कुछ भी।
जैसे मैं बहुत जल्दी थक जाती हूँ, कभी हार्ट में बहुत जोर से पेन होता है, बहुत ज्यादा, कभी चक्कर आते हैं काफी, कभी कभी धूप वगैरह में बहुत ज्यादा प्रॉब्लम होती है।
दिखता है तो वो नहीं समझ पाते हैं बहुत लोग। कई बार समझ में नहीं आता कि बताएं कि नहीं बताएं, कि बताने के अलग प्रॉब्लम है और ना बताने के अलग प्रॉब्लम। तो जब से मुझे ये मालूम पड़ा है, मेरे को यही है कि ये टेस्ट हर जगह, चाहे प्राइवेट हो चाहे गवर्नमेंट हो, कंपलसरी होना ही चाहिए। जैसे जैसे भी प्रेगनेंसी में जो सब रूटीन टेस्ट होते हैं उसमें ये टेस्ट जरूर होना चाहिए। अगर आपका एक मतलब एक तो कॉस्ट भी बहुत ज्यादा है, दूसरा उसका मैच मिलना बहुत मुश्किल हो जाता है, तीसरा मिल भी जाए तो
सही होता है।
हाँ, उसके बाद फिर उसके रेशियो जो रेट है सक्सेस के वो काफी से वो डॉक्टर बताते हैं कि 6।
So just sharing a few questions and experience just led me to believe that, they were trying to live their lives. They're being very positive about it. But the questions remained that why did we not know about this simple test before conceiving? Why did a simple test was not advised to us? I mean, this was, and they said that I take every opportunity to tell everyone that जैसे कि आप hemoglobin कराते हो, क्या आपने ये test कराया? So that was the kind of trouble this lady was in. And the girl was so smart, the girl was so active, but every 25 days she was receiving a blood transfusion. So I think it was so moving to meet the parent and the daughter that, but that is something which is completely preventable. So with that, I would like to invite Dr. Madhulika ma'am to talk about the prenatal diagnosis and, how we can prevent it and, how we should go about it.
Thank you, Aparna. Thank you, Dr. Khurana, and the whole team of SFM for inviting me for this very, very important session. So, Ratna has covered quite a lot, which is required for understanding about the prenatal testing. So, she has already told you about the mutation testing, and then how do we screen the families, and then if husband is negative, but then she has already told you that we have to be actually we have to be really cautious about it, being the husband being a silent carrier or any other complexities because of the presence of other modifying hemoglobins. So basically, and thank you, Aparna, for putting these slides together as well. You can move to the next slide. So basically, these are some of the common mutations which we, so fortunately, there are 5 common mutations in the Indian population, and places where sequencing is not being done, ARMS is being used, so more than 95% of cases are being identified. So that is one important thing and good thing about thalassemia, that it's easy to identify the mutations. You can go to the next slide, Aparna. So, so basically, prenatal diagnosis of thalassemia is being done. See, it was probably the first disease for which prenatal testing was made available. And when Dr. I.C. Verma established this unit, it was very early that it was our lab and lab in Mumbai were the first labs where prenatal testing was offered. So basically, nowadays, we are focusing for prenatal diagnosis, we are doing a CV biopsy, and the earliest time is, you all know, you're all fetal medicine people, 11 to 12 weeks, but it can be done at any gestation for that matter. We also use amniotic fluid, which has not been put here. So amniotic fluid is also a good source for doing mutation testing for the fetus. And typically we do it after 17 weeks because we need to get good amount of cells to sort of extract the DNA. In the table which was put by Aparna, she has also put cord blood HPLC, which we use very, very rarely these days, and it was a time when sequencing was not being used routinely and we were stuck with testing for only 5 mutations. In those situations, cord blood HPLC was being tried. The reliability of this test is not very high. And the more important thing here is that there can be maternal cell contamination. So we would very, very, very rarely at this point in time resort to cord blood sampling. So CV biopsy we prefer, and then we can also do amniotic. You want to show the video, Aparna?
Ma'am, that's
Okay,
fine.
so yeah, so the basically what is being done, I would just like to give you some take-home messages that Ratna has already very clearly told this, that when we need to do a prenatal testing, and then based on gestation, we choose chorionicolous biopsy or amniocentesis, and rarely we would do a cord blood testing. So there are certain prerequisites which also Ratna touched upon. Maybe they're there in the next slide, Aparna. So one thing which is very important when we are offering prenatal diagnosis is that it is good for the monogenic disorders like thalassemia that we can clearly give them a risk of recurrence. And I think this is very important as the family needs to understand that risk of 25% is in each pregnancy. Even now, despite explaining many times a couple things that it will be 1 of 4 pregnancies. So if one is affected, three next are going to be. So this chance does not have any memory. We have to explain this to the family. Confirmed diagnosis is extremely important. So we would usually do not take a prenatal sample unless we have a confirmed molecular analysis on the affected child and parents. Very rarely, if they have come late and we have no doubt about the diagnosis, then at times we take both the mutation testing from the family and the CVS together. Now, as I said, without having a molecular diagnosis, we would not take a CVS. And CVS we would always prefer for monogenic disorders because they have a good yield of DNA. But as I said, amniocentesis is also a good sample, and we very often use it for prenatal testing. So this again, Ratna has already shown, and this is just to show you that with the sequencing coming in the majority of patients, we are able to identify the mutation. So we have seldom resorted to nowadays to doing a cord blood HPLC. Next slide. So you all know that we have to counsel before doing an invasive test and explaining about the disorder. So procedure-related risks are to be explained, and you all know that it is a learning curve, but in experience centers, the procedure-related risks are very, very low, say something like 1 in 500, and it's almost equal for amniocentesis and chorionic villus biopsy. One thing which I would like to emphasize here is that apart from the procedure-related risk, we have to explain them the risk of the test being wrong. So maternal cell contamination is one of the important risks because of which you can have some error rates. So overall, when you see the error rates are very low. Overall, if you see that. So in experience centers, we hardly have wrong results. But then maternal cell contamination, particularly in chorionic villus biopsy and FBS, we usually do not do in amniotic fluid. It has to be done before we give the report, and you should make sure any report which you are getting from any center should also have the report of maternal cell contamination, because that is obviously going to give you a wrong report if it was all maternal tissue and no fetal cells. Next slide. So when for maternal cell contamination, basically you have to visualize these villi in a petri dish and very, very carefully dissect these villi under the microscope. And experienced people would be able to remove the residua from the villi and then have a pure fetal tissue to do the prenatal diagnosis. Next slide. So this is a must in all situations when we are doing a prenatal diagnosis, particularly using CVS and cord blood. And there are various methods of doing it. I'm not going in the details. So a molecular method using markers and qFPCR. And this next slide. So just to show you about what we have been experiencing over years. So typically, we have been doing 80 to 100 prenatal testing every year. Now the numbers at times in some years is lower because many more centers are also having prenatal diagnostic facilities. So just to sort of review our data, this is old data. So out of say around 1,500 deliveries, out of say these many deliveries, about 24% were affected. So this sort of tells us that the results are reliable. So 25% fetus is being affected. So, and the similar experiences there from other centers as well within the country. So we have very robust prenatal diagnostic facilities in the country, but then I think we need to have more because there are still less to cope up with the numbers which we have right now. Next, I don't know if there's anything else. Next. So Ratna has very, very nicely alluded to that when prenatal diagnosis is not required. So this slide I have also put just to re-emphasize that many times we are still getting referrals with one partner being carrier for prenatal testing, and then all these combinations of HBD and THAL and HBD and E and so on and so forth. So in these situations, there is no need to offer a prenatal testing and only counseling is required. So I think that was all which I wanted to say, and I would be happy to take any questions. Thank you, Aparna.
Thank you, ma'am. Thank you so much for clarifying all of this. And I think the data that you also showed is really nice that, 25% of the babies are, usually affected. And we really require this kind of robust testing, and many more facilities are required. And we are doing around 80 to 100 CVs per year. So I think that's what we wanted to know. So thank you very much, ma'am. And if there are any doubts, I think we can clarify them towards the end. So thank you very much. So just going to the next part is about the blood transfusion. So how do we manage and how can we make it safer? So I would like to invite Dr. Haim that, what are the basic principles and how can we make it safer for these thalassemics?
First of all, thank you to Dr. Aparna and SFM for inviting me to this panel. Giving transfusion to any patient is a very difficult decision. So in thalassemia, they are going to require the transfusion for lifelong. However, once a decision to transfuse is made, we have to first think about few things which needs to be done before transfusion started. First of all transfusion, despite whatever testing is being done, carry a risk of transmission of certain infections. So we need to have a baseline of some viral infection like HIV, HBV, HCV. Apart from that, we need to have immunization of the child done if it is not already done. Hep B is quite prevalent in our country, and there is around 1 in 60,000 transfusion to transmit this infection despite NAT testing. The second thing is to identify the blood group as well as to have an antibody screen. Some conditions have naturally occurring antibodies which we need to see so that they don't interfere in our future testing. We need to have a phenotype of the child if it is not already done. If it is first transfusion, we need to have it done at that point of time, because once the transfusion started, they are going to start for lifelong, and then serologically, at least it is impossible to see the phenotype of the patient. And molecular techniques for blood grouping are very limited in our country. So we need to do a serological phenotype initially, and we need to add that just to ensure that that does not interfere later on with our testing. As far as blood components are provided, we should not go for whole blood. Most of the blood centers now have blood components, so we should go for packed red blood cell. They need to be within 10 days. In most of the settings, sometimes it is not possible to give for some blood banks, but ideally they should be within 10 days to 14 days. They should be leukoreduced. That is one of the important part. Leukoreduction may be by Buffy-Coat method or by proper leukofilter. So if you are using Buffy-Coat method, they will just prevent the FNSTR. So if a patient is having FNSTR, that might be prevented using Buffy-Coat method to leukoreduce. However, the ideal way is to do through leukofilters. They will, in addition to preventing the FNSTR, they will reduce alluminization to leukocyte antigens in case the child opts for BMT and a suitable match is found. They will reduce the risk of transmission of CMV, and that again would help in the future BMTs. Then the blood which is to be given has to be identical. Most of 99% of the cases, it has to be identical. Sometimes if there are alloantibodies, then we need to give compatible blood. And if phenotype is known, then every effort should be done to give phenotype matched blood. If alloantibodies are identified, then we need to give unit which is negative. So phenotyping is important, and phenotype matching is important to prevent alloimmunization due to the transfusions. And one has to ensure that the testing is done by high quality assays, because if it is not high quality, if it is not NAT, there are high risk of transfusion transmission of virus infection. Then the transfusion therapy has to be maintained above 9.5 or 10 grams. So whenever a child hemoglobin comes down to 9.5 or 10, we need to start transfusion. And we need to see that the typical post-transfusion hemoglobin is above 14 grams. Usually it returns to baseline in 3 to 6 weeks, depending on child to child. However, and this is a rough guide to identify how much blood to be given. So ideally, the blood centers usual packed cells have a hematocrit of in between 50 to 70. So if you follow the 60% hematocrit rule, you will be transfusing almost adequate amount of red cells to the child. So if your target is to improve by just 1, you can give around a rate of 3.5. So you have to decide according to child's weight and the target hemoglobin, how much you want to transfuse. So average 60% if you are taking into consideration, you'll be transfusing an adequate. Next slide. Then initially you need to decide how frequently to transfuse. So ideally, initial schedules are at 3 to 4 weeks interval, and the target is to maintain hemoglobin above 9 to 9.5 grams. So before next transfusion, we need to evaluate if the hemoglobin is below the target. That means the first transfusion, the previous transfusion was inadequate. So we need to give more frequently every 2 to 3 weeks, or we need to increase the volume of transfusion. And we need to have a target of around 12 to 12.5 in between these transfusions. And it should not increase more than 15 grams per day. So this is for a child where there are no complications. If there are cardiac complications, the hemoglobin level might change in those patients. Next one. And while doing transfusion, we should not forget about a lot of complications which may occur, like hemolytic transfusion reactions may occur due to alloantibodies, alloimmunization may occur. So there are some specific thalassemia patients like thalassemia intermediate group where even a single transfusion can form multiple and rare antibodies. So we need to be very careful when transfusing the next time. Before every transfusion, antibody screen should be done, and we should be very careful while transfusing these patients.
So thank you, Dr. Haim. I think so. Dr. Haim is our blood transfusion lab specialist, and we are working together in a lot of things together. And he is very, very passionate and helps us really a lot. And I think he's got experience of helping these kids a lot. And I was asking the cost of transfusion. Do they need to donate very often? So if there is a person who's thalassemic and he's registered with the blood bank, do they need to donate blood frequently?
The amount of blood which they require, I think I also won't be able to bring so many donors. So we don't ask thalassemia patients to donate blood. However, we usually tell them that their parents can donate at least once in a year. So they will understand what donors undergo when they are donating blood. So, and we are deficient in blood, most of the blood banks in India. So we don't force, but we ideally tell them to donate. There is no necessity to donate. Ideally, the blood bank would arrange blood for them. Secondly, there are no costs involved in thalassemia. In our country, only the cross-match charges can be taken, but not the actual cost of the blood unit. So that is something which government has ensured that no blood bank can charge for the blood unit. Only for cross-matching charges they can take. That is the government's policy.
So thank you, Dr. Haim, for making that clear, because I was talking to 2 or 3 parents whose children are thalassemics, and they were saying that, some of the blood banks, they do ask for parents or relatives to donate once in 6 months. And they had real difficulties during COVID times because they would not find anybody. But I think that really varies from place to place. So thank you, Dr. Haim, for joining. And Dr. Mukul, can you summarize the treatment of thalassemia for, what are the ancillary treatments and the newer drugs and the current status on the bone marrow transplant?
Thank you, Dr. Aparna and Society of Fetal Medicine for inviting me for this. So thalassemia management is like a multidisciplinary team management. We have to understand thalassemia as a spectrum of severity, where thalassemia major or transfusion-dependent thalassemia patient who require transfusions every 3 to 4 weeks, to a carrier stage who require transfusion very, very infrequently or during illnesses only. And there is a intermediate kind of a thing which we previously used to call thalassemia intermedia, who require transfusion intermittently or at a higher interval than the thalassemia major, maybe once in 2 months, 3 months, or during pregnancy or illness otherwise. We need to realize that in this subgroup of patients, the thalassemia intermedia, there are certain complications which are more commonly seen, like gallstones, like extramedullary hematopoiesis, leg ulcers, and thrombotic events. This happens because in thalassemia major patients, we are suppressing their inherent erythropoiesis or the abnormal red cell production by giving regular transfusions. But because thalassemia intermediate patients are not getting these regular transfusions, so these abnormal red cells are circulating in the and they cause, by various pathogenic mechanisms, they cause these complications. Vis-a-vis thalassemia major patients, because of their chronic transfusions, they result in iron overload and subsequent endocrinopathies, which I will discuss further. So what happens is, actually the treatment for thalassemia at present available almost in many of the centers across India and across world is stem cell transplant. So this is the only curative treatment available at a reasonable cost to us. The other curative modality which is now available is gene therapy, which is prohibitively costly even in the West. So what we have, I'll discuss slightly about stem cell transplant. So this is the curative modality offered in many centers across India. So what it gives is the freedom from blood transfusion and associated side effects after some time. What happens is you have a HLA match sibling donor or a match unrelated donor from whom stem cells are collected from the blood or bone marrow. These stem cells are processed or cryopreserved. Meanwhile, the patient is giving chemotherapy to suppress their bone marrow and enough immunosuppression so that the stem cells from the donors can harvest over there and grow over there. These stem cells are infused and it takes around 2 to 3 week times for the patient to start where these stem cells start the production of the donor blood. And the patient takes around 2 to 3 weeks to get to the stable kind of blood reports. So the success of this process, it depends on the age of the patient. So lower the age, better it is. Usually, the thalassemia transplant are done below 10 to 15 years of age. It also depends on the iron and the liver status of the patient. So if the patient are well chelated, they have a low ferritin level, they are better, the outcomes are better. The chances of complication increases if their ferritins are very high or if they have hepatomegaly and associated liver fibrosis. It is said that early transplant in less than 10 years of age in a well-chelated patient gives best outcomes. So there are some immunological and infections complication which can occur post-transplant and which can be a limiting factor. The good thing is that the government of India through Coal India program, it offers around 10 lakh rupees support to around 7-8 centers across India uh, via a national program under CSR initiative. And many of the centers, some government and some private centers are using this facility to transplant majority of the HLA match sibling donors. So at our center, it costs around 10 to 15 lakh rupees. In a private hospital, a sibling transplant will cost around slightly more than this. Next, I come to blood transfusion. So Dr. Haim has beautifully described about blood transfusions. We just remember that each unit of blood, it comes with around 200 milligram of iron. The problem with iron is once it goes into the blood, it cannot be excreted either in the urine or in the stool. So all chelation, that is, these are chelations are agent which bind to this iron and are created outside of the body. So the indication to start chelation is usually after first 10 to 20 transfusion when the serum ferritin rises to more than 1000 milligram, or usually after the patient is 2 years old in a transfusion-dependent thalassemia. So there are two drugs, oral drugs, which are available, deferasirox and deferriprone. Deferasirox is the most commonly used drug nowadays and is a very effective chelator. And for kind of refractory patients to these or who have a persistent iron overload or severe iron overload despite oral drugs, we go for a parenteral desferrioxamine drug. So if the patients are planned for transplant and in the post-transplant period, one thing is that the patient are to be given irradiated blood products, which is kind of essential in these patients. One of the interesting things which occur in non-transfusion dependent thalassemia is even though they get very infrequent transfusion or maybe every 3 month or 6 monthly transfusion, they kind of have ineffective erythropoiesis which is going on in their body, and some with different mechanism it leads to increased absorption from the intestine. So despite having very infrequent transfusion, they may end up with iron overload complications. What complications can occur? Complications in the, this iron can deposit into the liver, heart, and various endocrine organs and cause damage to these tissues. So if in the liver, it can cause a hepatitis and ultimately can lead to liver fibrosis and cirrhosis. Cardiac iron overload in a long term, if severe, can lead to various myopathies and arrhythmias occasionally. And endocrinopathies are again very symptomatic in these regards. So, particularly endocrinopathies can occur in the pancreas, in the pituitary gland, thyroid gland, and gonads. So, these patients are to be monitored for these complications. There is a question about role of splenectomy. So splenectomy usually is not required in thalassemia, well transfused and well chelated thalassemia major patient, but occasionally patients of thalassemia intermediate, they present with very large spleens or worsening anemia, leading to when it leads to poor growth or development, and when the patient have a large spleen with symptomatic cytopenias. In such patients, splenectomy can be done, where it improves hemoglobin by around 1 to 3 gram%. Splenectomy can be associated with infection risk, especially in very young patients, and with risk of some risk of thrombosis in a longer term. Then there are certain drugs which to be given in thalassemia intermediate or non-transfusion dependent thalassemia. So hydroxyurea is one such good drug which is given to almost all our patients of non-transfusion dependent thalassemias. Usually starting at a lower dose of 10 milligram per kg and titrated upwards to maintain a good neutrophil and platelet count. So it is able to raise hemoglobin by 1 to 2 gram% at 6 months. So it's a pretty kind of a good drug. There are two new drugs which have been in use. So one of them is luspatercept. It acts by stimulating erythropoiesis, and this drug has been approved for patients who are more than 18 years of age. It's a subcutaneous injection given every 3 weeks, and leads to slightly decrease in the transfusion requirement and transfusion independence in some of the patients of thalassemia major and intermediate. Uh, there is another drug that is thalidomide, which is given to patients as a HPF inducing agent, and it has been tried and given to many patients, but across India and across the world. So the rates of response uh, measured in the terms of decrease in transfusion or even transfusion independence, they have varied from around 25% to even 50-60% between different studies. So even though not approved, but this is one of the commonly used drug nowadays for transfusion dependent thalassemia and non-transfusion dependent thalassemias as well. There are certain other complications like gallstones, which can occur, for which cholecystectomy is advised. And occasionally, especially thalassemia intermediate patient can have extramedullary hematopoiesis, which can occur especially around spine or nodes. For these patients, steroids or hydroxyurea or surgery is advisable. Complications like thrombosis can be subclinical, all clinical, especially increase after splenectomy, and these are to be monitored for. Just remember that overall, what I want to emphasize is that comprehensive thalassemia care is a multidisciplinary team effort. So you require a pediatrician or a hematologist at the helm, and accompanied by a hepatologist, cardiologist, endocrinologist, genetic counselor, fertility specialist, social worker, nurse specialist, psychologist. So many people are involved, which are managing the patient. And in addition, like we have discussed, the important role of prenatal diagnosis and other things. Lastly, I just want to say about gene therapy. So this is something which is now FDA approved and available in the West. So it's a good thing that this is being available. At present, the cost is around $2.8 million for a patient, which translates to somewhere around 2 CR for our patients if they go abroad. It's still not available anywhere in India at present, although there are certain, trials which are being in the pipeline for these with government support, obviously. So hopefully in the future, we will have gene therapy options as well available to us.
Right.
Thank you.
Thank you, Dr. Mukul, for putting all of it out there. And I think with good management support, and that is why we are seeing more and more women with actually pregnancies with thalassemia major also. We do get to manage these pregnancies, and that is the reason why, because we do have now comprehensive multidisciplinary management, which are leading to, pregnancy, women or general population reaching adulthood. And so thank you for comprehensively telling us all the management option and putting all of these things out there. So thank you very much. And if there are any questions, we can take them later. Thank you very much.
Thank you, ma'am.
So now, ma'am, Dr. Sumita Ghosh, ma'am, you've had to wait for a long time, but I think we are all waiting to hear from you. We've heard you talk about a lot of things, but I think we want to know, like, when we put across public health strategies, we want to have strategies that work. We do understand that this is something which is needed, but how to make a balance, and how that to make an effective public health strategy. What has been your experience, and how do you think that this should be taken forward?
Thank you, Dr. Aparna and the SFM for giving me this opportunity to speak on Thalassemia Day. Huge public health problem with huge prevalence, with a lot of resource intensive management, financial burden on the family, the psychosocial impact of the person who is suffering, and the parents. So from every aspect, it is a public health issue of great importance, and it must be taken up seriously. But having said all this, the public health goal, the policy has said that, prevention to reduce prevalence. That is the first thing, through various kind of activities which are sustainable, which will cost effective, and which will give results. Empower prospective parents to sort of exercise their right not to have a child with serious genetic disorder. So that again is a very important aspect, that they have access to family planning, they have access to NTPs if they are knowing that this child is going to be seriously affected. Then empower an affected child to have access to optimal care. So optimal here, we know that this management, ultimately it is a lifelong process and needs a bone marrow transplant or higher managements. So optimal management, have been defined under public health so that they have access to the basics. So the goals, measures, there are various measures which have been taken to reduce the prevalence. The first one is awareness and education programs, especially at the adolescent age group, at the school health age group. That has been a policy decision. Screening of pregnant women, that again, there is a guideline, NC guideline for thalassemia screening. The issue here is that it has not been taken up by many states. As you know, central government and state work hand in hand with our public health programs. So the states have to demand that this should be done. So in states where it is high prevalence, so there it is happening. Premarital and preconception carrier screening, that is a very important step, as has been discussed already, that if we know the carriers, and you can, before marriage also, we can give counseling, and during marriage, we can have some sort of prospective groom, we can do some kind of screening. When the pregnancy screening says, a lot have been said about it, that she is a carrier and the husband is also, and the couple is at risk, then genetic counseling and also family planning counseling. These are part of our policy. Having said that, we must do screening. That is very important, pregnancy screening and even pre-pregnancy screening. That is very important for prevention, and many countries have shown that reduction can be almost to zero birth of thalassemia babies. But again, the facilities, public health facilities for labs for such screening of this 3 crores of cohort, pregnant cohort, that is again an issue. So that is a policy how to strengthen. And at medical college as well as district level, efforts are on to do the prenatal diagnostics and newborn screening. This is a very important issue, and it has been touched upon, that RBSK. RBSK is a program which is done in schools and Anganwadis. But under RBSK, there is a program for newborn screening, which happens at the delivery points. Basically, not everywhere thalassemia is being screened, but when the states, they want thalassemia screening, congenital, during the newborn period, like Delhi has actually, they want it. So that support has been given. So we heard that it is happening, but there is at present, because sustainability an issue. So now it is, staffs are not there or something like that. We heard, but it was supported under NHM. Then adolescent students, there is a effort. There is an effort to, do the sickle cell screening. Actually, sickle cell mission has been announced last year, and under that, a lot of screening has been point of care and all, and lot of capacity building has happened, and lot of these lab setups, screening diagnostics have been improved upon. So we think this thalassemia screening also will ride on the back of the sickle cell mission, and we'll see in very near future we can see a major jump in screening. Other things which are there is free admission for the thalassemic patients, blood transfusion, iron chelation, even at district level, chelation therapy is kept. And during sickle cell mission, it was discussed that depending on the load of, how many, medicines will be required, the even the block level, sub-district level can indent from the district level. The PMJY, as you know, Ayushman Bharat PMJY, the insurance scheme, it has included whole blood transfusion as well as blood component transfusion free of cost to the eligible BPL categories, those who have those who are included in this. The strengthening the component, blood giving, the infrastructural requirements, again, it is being supported in public as well as some charitable sector. Blood banks are being supported from our blood cell division in the NHM. Counseling has been, included as a part of management because of the very serious issues they have. In March 2024, the permanent disability status have been, accorded to thalassemic babies and benefits thereon. There are some efforts at setting up Center of Excellences at tertiary care centers. New RBSK again is a very important program because newborn screening, if it is missed and if they are at school, they are detected, they are referred and they are taken up for, and the reporting is also done. That reporting has been made, mandatory, that if thalassemia new cases are detected, they have to be reported at the district hospital. Various facilities have been strengthened, as has been said. Thalassemia Bal Seva Yojana, which is ministry has taken as a CSR initiative with whole India, and 10 lakhs are provided to eligible patients. And till May 23, I have the figures that 356 bone marrow transplantations have been done. Major activities: information, education, awareness about various things, and especially inclusion in school curriculum. We have recently we have asked NCERT to include it in this. Various media and other this thing, and in a collaborative approach, center is supporting IC activities on thalassemia to the states and various organizations. Next. So having said all this, management and detection, they have serious challenges. The data, epidemiological data is totally, not with us. It is incomplete, and the precise burden of this disorder is unknown too, because it is silent for the carriers. So again, though they do not have symptoms, but they have the potential to be transmitted to their children. So that is the issue here. General awareness is lacking about counseling, and the expertise to do the genetic counseling, as we saw that the complexity of the situation, how who can be, have the child, who cannot have the child, where the probabilities are more. So these kind of giving that counseling is very, very, that expertise is not there. And also the, demand is less. The demand supply gap in care, access of care, in spite of all the efforts they still exist, especially in the remote areas, where they have to travel every 2 weeks. Where will they travel? Like how will they travel? So that is the financial burden, the psychosocial impact. And we heard about the various complications, challenges in treatment, in the disease complication, the treatment complication, infections, and these are, basic challenges. So I heard the question, why not it has been made mandatory under RBSK. RBSK, it has not kept in the optional list because not many states opt for this. They only, when they think that this is a real problem they are facing, they opt for it. And center state support in health is, when they demand, we are providing. So that way it has not been made mandatory because of the resource intensive nature of the disease. And there are, of course, research gaps, therapy gap, because we need some easy technology, cost effective technology. Like sickle cell, we had point of care tests, so we went ahead with it. So that kind of, because feasibility, sustainability, infrastructure building, all these are very important for one thing to be taken up in public health perspective. But serious efforts are on for prevention as well as for, so that they get some access to care, and also the parents have a choice. So these three things we are working towards.
Thank you, ma'am, and thank you very much for bringing about what is being already done, the efforts which are going on, and also the challenges. And I think the challenge of thalassemia, as you really said, is the complexity. And the webinar really shows that we've had the obstetricians talking today, we have the fetal medicines talking today, we have the public health expert, and we have the hematologist, and we have the blood transfusion experts. All are talking about one part of thalassemia, because everybody is really passionate about one part of thalassemia. But unless everybody of us really comes together, this is not going to go away. So I think that is the link, that everybody has to be linked from one part to the other, and that is what the complexity of the whole situation is, and that is why thalassemia is really still existing. So just to end, to say that I recently contacted, for the occasion of Thalassemia Day, Mrs. Alka. She's a thalassemia warrior who actually, she's now around in her middle age. She's she's a thalassemia patient, and she now advocates for greater awareness and support, and understanding for those living with thalassemia. And she is actually a president. She won a president's award, and now she works with the in PGI Chandigarh with organ donation. So it is not that I had a video recorded for her, but I think it is already 10:15, so I will not share the video right now. But she shared her story that from the very childhood she had such difficulties, because it was a time when blood transfusion was not so easily available 35-40 years back. And but the technicians in PGI, the father of my colleague who actually shared this video with me, really went out of his way to arrange blood at that time. And through her struggles, the struggle for blood, struggle for chelation, she never lost hope. And now she provides hope to so many other people. But yes, a small test, a small prevention, a small step towards awareness can improve the lives of so many others. So lives of people like Alka can actually be a torchbearer for what all can be done if we do have the right approach to prevention and awareness. So these are some of the organizations which are working towards prevention, which are working towards providing treatment, helping with the treatment: Thalassemics India, the Thalassemia and the Sickle Cell Society of India, who are available to help in case that is required. So I would like to end by saying that hemoglobinopathies are one of the major public health problems in India. To achieve success in their prevention and control, an ongoing holistic approach is required. It is expected that with the optimal collaboration and support, effective prevention and control of thalassemia can be achieved. And this will lead to a healthier new generation which enjoys an overall better quality of life. And as Dr. Radhika said, that as we are moving from mortality and morbidity to a better quality of life, there is no reason why we should not start thinking about screening, awareness, and one simple test. And like that Preeti patient told, Preeti, the mother of Pragya, said, it is one simple test, and why we were not doing it. So I think this is time that we really look and say that we should do HPLC for all patients in pregnancy. At least I think for the Society of Fetal Medicine, one message, if we want to think about it, we would like to say that, at least HPLC can be done for all patients who can afford it, and we should be advising it routinely. So with that, I would like to thank our esteemed panelists today. It's been a great discussion. All of you have done immense amount of work in thalassemia, very passionate about the work that you're doing. Thank you for being here today and sharing your experience. Thank you very much.
Thank you. Thank you, Aparna, for this. We actually have some very brief questions in the chat, and I was wondering whether we can take 5 minutes for those.
Yes, sir. I think let's quickly take those questions.
Yes. The first one was a suggestion by Dr. Chelapan, who thinks that every lab should have a biorad machine for HPLC. It costs less than 10 lakhs and the sample cost is then about 300. And you can manage both HbA2 and HbA1c. So you can actually screen for two diseases at one cost. And that we really can actually consider this. So I think that's a very good suggestion. And then he also has said that in the district in the tribal belt, there's a high incidence of sickling. And there are school children screening is being done and is well accepted by these remote tribes. So it's very encouraging, really, that is available. And so it's not just that, it's not just the urban population, but that the rural population will also be quite happy with this kind of a thing, which is very nice. And he also had a suggestion that maybe that in the MBBS curriculum, we may need to hammer this into the candidates, to use his own words, to interpret the indices so that we can understand this. And I'm sure Dr. Ratnapuri can put a bit of light on this. Is there any, truly speaking, where is genetics fitting into our MBBS curriculum today?
Well, actually, there's been lots of people have been working with the government, with the organizations to try and put it, but I'm not sure that it's there as yet. I don't know more about that, Dr. Madhulika ma'am. I don't think it's worked. We've given any suggestions.
Yeah. So they're trying to incorporate it in the UG and in the PG curriculum, but then it's being worked on for a very long time. Suggestions were taken from all of us also, what all to be integrated, but it's not really well in place. But definitely it should be included in the curriculum.
Yeah. Dr. Chelapan also wanted to know about the role of alpha globinopathy in couples with raised HbA2.
If there is an alpha gene mutation, normally the hemoglobin A2 is normal. There is microcytic hypochromic anemia. There's no iron deficiency, and the A2 is normal. It's not raised.
And there is one question, and this is from Dr. Asmita Singh. Ma'am, if couple presents late at 12 to 13 weeks, can we directly do prenatal testing without knowing the mutation and molecular testing?
So, Asmita, what I said during the discussion also, that we prefer doing confirming the molecular, doing the molecular testing and confirm the mutations. And at 12 and 13 weeks, it is not really very late. Most of the time, we will be able to get the mutation report within a week or so. So we can easily do it. Now, I mean, not that we need to sort of prolong it or make it late, but then now the limit is 24 weeks. So 12, 13 weeks is quite safe to get the mutation done first and then do the prenatal testing.
Wonderful. So we know exactly what thing to do. We have a couple of minutes. I think Dr. Chalapan has his hands up. I see his, can he be made a co-host to be able to ask that question?
There was one question, Dr. Khurana, about how do you test for silent carriers? Maybe Ratna can answer or I can answer. So the
Yes.
question was that do you do HBB gene sequencing to know the silent carrier status? So that's true. And there are certain mutations which have been found to be typically associated with silent carrier status. So that's another question.
Excellent. I think that completes the whole series. Is there
Ma'am, I
any-
just want to put it in the correct perspective. I read it because she said for a couple. It's very
Okay.
rare to find a
Yeah,
couple
yeah,
who's a
right,
silent carrier.
right,
So we
right,
will
right.
not start sequencing everyone.
No, definitely not.
If one
Definitely not.
partner is a
Yeah,
carrier,
yeah,
then
definitely. If one partner is a carrier, and even like at least as was being suggested that as you also said that the recommendation is that you should, even if the HbA2 is normal, they recommend that should sequence. But most of the time, what we will do for borderline cases is we will look at the indices and other things and then decide and the family history, of course,
yeah.
decide about sequencing. Yes.
And where they come from, I think that also
Yes,
makes
that's
a lot
also
of
very important.
sense, which part of the country.
Thank you very much. That brings us to the end of this evening. Any last words from Dr. I see Dr. Bimal Sani is still with us. Bimal, you've done a fabulous job for our outreach programs. How do you think we should incorporate thalassemia into our outreach programs?
I think that should be the buzzword this year now.
Yes.
We definitely need to, I think, all these things which can be prevented, that should be the buzzword. And, why outreach? Because that is where you have to take it eventually. So why not right from the beginning, see to it that, all these smaller, everybody everywhere becomes aware of that. And unless we don't do that, we won't be able to prevent this. And fabulous, the whole program is, it's an eye opener, in fact, for quite a lot of us. And great panel, great lecture by Dr. Ratnapuri and everyone. And I just feel that somewhere down the line, that feeling comes. It's such a simple thing, why we have, why we really had to wait for such a long time for really bringing this up. And I think we'll be very soon, thalassemia screening would be a basic part of, in the protocol for every pregnant woman, definitely.
I think if we have one member in say, every district of the nation, then we could just put all our ideas together. So I was wondering whether Dr. Ratnapuri can put together a committee with just one person, and then we start advocacy with the government. And just like we managed to find Dr. Ghosh is such a wonderful person to put forth our MTP Act, which really was one of the greatest challenges for the medical profession, and it was only with her support that we could achieve this. I was wondering whether we could give a thrust to our thalassemia program in the same way. And once we have a representation from every single district in the country, which I don't think is impossible for SFM, perhaps she can push her agenda just the same way as she was able to push initially the guidelines for late terminations and then the actual act itself.
Can take it for me. Can take it.
Yes. Wonderful.
Thank you.
Excellent. Well, thank you everyone for being with us tonight. And on behalf of Dr. Mohit Shah, who had to leave in between.
No, I'm here, sir. I'm here, pretty much here.
Oh, you are there. Mohit, I don't see you on the main panel. Please go right ahead. Yeah.
No, this entire thing has been such an eye-opener. I mean, I really applaud the humongous efforts all the panelists are putting in. And I completely agree that we as an SFM can take this forward and make us maybe a small dent. Eventually, it should catch the momentum. And if we hopefully can one day eradicate thalassemia completely. But I agree that we should include the test in, at least we can percolate this fact that this test has to be included and offered to every pregnant patient. I think that's a job half done, I guess. But a humongous efforts, all the panelists, all the experts. I mean, I was amazed by the work that is happening in the field of thalassemia today. I really salute you all.
Thank you, and my special thanks on behalf of Society Field Medicine to all the panelists who came from such diverse fields and gave us support in taking this message across to our entire society. We're very proud to have had crossing our milestone of 5,000 members, and I think this was an ideal occasion to celebrate this. Thank you indeed, and good night.
Good night, everyone.
Good night, sir. Good night, everyone. Good night.
Good night.
Thank you. Good night.